S. Chan presents educational video lectures as per the new UGC CBCS curriculum. Absolutely free. Difficult science concepts made easy. Study anywhere, anytime.
Do you know what happens to your hair when you go for the rigorous hair treatment processes in spa and saloon? Sometimes you go for curling, straightening and many other things you do on your hair. What is the ultimate effect? What is happening actually chemically to your hair? Do you know that? Hi, I am Manika and welcome to S. Chand Academy, and today we will be talking about peptides and proteins, the important class of the biomolecules. If you want to study more about this topic, you can refer to our book by S. Chand Publishing, the link to which is given in the description box below. Let's start with today's topic, peptides and proteins. Peptides, as you all know, which discussed in our previous lecture also, that they are made up of the amino acids. So, basically amino acids are the building blocks of peptides, and peptides are thereby undergo combination to form proteins, right. So, basically the proteins are the biomolecules, peptides and amino acids are the building blocks. Okay. So, in this lecture we will be talking about sequencing peptides, that is how we actually know or decipher the structure of a protein. So, obviously that is a stepwise approach and we will talk about it in this lecture. We will also talk about synthesizing methods for the simple dipeptides, and we will take an overview of the proteins and their structure. So, सबसे पहले देखते हैं peptides होते क्या हैं. So, पहले lecture में अभी हमने देखा which was on amino acids. We saw that what are amino acids? I told you that they are bifunctional molecules and they have NH2 and COOH group. हमने कुछ reactions भी देखी which were common to almost all the amino acids, where I told you that amino acids behave like amines also, behave like carboxylic acids also. अब आपने ये चीज़ notice की कि अब किसी अगर आपके पास 1 mole compound है, it will have amino groups also and it will have carboxylic group also. So, it will always have a tendency that कि आपके जो कार्बोक्सिलिक ग्रुप हैं वो अमीनो ग्रुप के साथ जो है वो रिएक्ट कर सकते हैं, राइट. द कार्बोक्सिलिक ग्रुप्स कैन रिएक्ट विद द अमीनो ग्रुप टू फॉर्म व्हाट इज कॉल्ड एज एन अमाइड बॉन्ड. क्या कर सकते हैं? अमाइड बॉन्ड विद द लॉस ऑफ अ वाटर मॉलिक्यूल. वाटर मॉलिक्यूल के लॉस के साथ अमाइड बॉन्ड फॉर्मेशन कर सकते हैं. So, ये amide bond formation ही है which is responsible for the structural conformation of the proteins. ये proteins, peptides interactions basically are happening all because of the peptide bond formation, which is responsible for linking long chains of peptides and amino acids to each other. So, they are amino acids are linked together by amide groups called as the peptide bonds. अब amide group क्या होता है, उसकी properties कैसे होती हैं, क्या होती हैं, you can study about this from our previous lectures by S. Chand Academy only, right. So, during protein synthesis, the carboxylic group of one amino acid at the end of the growing polypeptide chain can react with what? Can react with amino group of another amino acid releasing a molecule of water, and the resulting bond between the two is known as a peptide bond. Very simple thing. Okay. So, this is how a chain of peptide will keep on growing. You will start with one amino acid, you will keep on adding another amino acid, another amino acid, another amino acid, so on and so forth till you get the desired peptide. Okay. So, in a peptide the principal chain contains what? The principal chain contains these amide bonds, and what is going to be the difference will be the R substituents. इसमें आपके substituents हैं जो differ करेंगे, and these substituent will determine the chain of the peptide or the peptide structure. Okay. So, individual amino acids which make up the peptide are known as the amino acid residues. इनको हम amino acid residues भी कहते हैं. So, in some proteins 2 or more polypeptide chains are joined together by डाइसल्फाइड ब्रिजेस। जब दो या दो से ज़्यादा पेप्टाइड चेन्स एक दूसरे के साथ बाइंड करती हैं सो वो कभी कभी डाइसल्फाइड बॉन्ड भी फॉर्म करती हैं अपार्ट फ्रॉम पेप्टाइड बॉन्ड्स। एक तो हो गया कि अमीनो एसिड्स किससे लिंक हुए, पेप्टाइड चेन से, और पेप्टाइड चेन्स आपस में किससे लिंक होती हैं, हाइड्रोजन बॉन्ड से, जो हम बाकी अभी प्रोटीन्स में पढ़ेंगे। बट पेप्टाइड चेन्स कैन ऑल्सो लिंक टू ईच अदर सिंस वी आर टॉकिंग अबाउट द कोवेलेंट बॉन्डिंग over here, we are not talking about electrostatic interaction right now. Okay, so another type of the covalent interaction in bonding which can happen is between the the thiol groups which are emerging from the amino acids which contain the SH group, यानी कि सिस्टीन अमीनो एसिड जहाँ पे भी होगा पेप्टाइड चेन में, सो दे हैव अ कैपेबिलिटी ऑफ अंडरगोइंग ऑक्सीडेशन। सो बेसिकली जब थायोल ऑक्सीडाइज होता है तो डाइसल्फाइड बॉन्ड फॉर्म हो जाता है एंड ऑब्वियसली इट लीड्स टू more and more branching in the polypeptide chain. So polypeptide chains can be linked together by disulphide bridges. Okay, let's talk about the nomenclature, how we name peptides. अब आपने देखा हमने जब डाइसेक्राइड्स की बात की थी प्रीवियस लेक्चर में, कार्बोहाइड्रेट्स जब हम स्टडी कर रहे थे, आई टोल्ड यू दैट I talked about that to name a disaccharide we take the first parent carbohydrate, first hemiacetal sugar as the parent carbohydrate, उसके बाद जो भी आपका सब्सटीट्यूएंट है उसको आप एज अ पेंडेंट ले सकते हैं। इसी तरीके से पेप्टाइड्स का जब नॉमेनक्लेचर करते हैं तो दे आर नेम्ड स्टार्टिंग फ्रॉम द फ्री अमीन एंड ऑफ द पेप्टाइड, उनकी नॉमेनक्लेचर हम फ्री अमीनो एंड से स्टार्ट करते हैं एंड एज द नेम्स ऑफ द अमीनो एसिड्स आर जॉइंड टुगेदर then the each next amino acid is considered as a substituent for the following amino acid. जैसे ये एक peptide chain है, here you have a phenylalanine group, then you have an alanyl group and then phenylalanine, leucine and threonine, phenylalanine, leucine and threonine. So this will be named as phenyl alanyl leucyl threonine. Okay, so this is all based upon threonine. Okay, आप नेम करना कहाँ से स्टार्ट करोगे, फ्री अमीनो एसिड से, एंड द पेरेंट विल बी द लास्ट अमीनो एसिड, दैट इज द अमीनो एसिड प्रेजेंट एट द कार्बोक्सिलिक एंड ऑफ द चेन. So for example, bradykinin, okay, is named as this. It's named as a derivative of arginine because arginine is the last amino acid in the chain. बाकी सारे are taken as the substituent without any spaces. Usually name में spacing नहीं होती, जैसे आप यहाँ पे देखो there is no spacing. So this is how they are named. But yes, if you are writing the 3-letter abbreviation in the writing the peptide sequence, then you have to give a spacing with the. So writing a peptide sequence, जब भी आपको peptide sequence लिखना होता है, suppose it is given to you that you write the peptide sequence using the 3 amino acids, say A, B, C. So by convention, the peptide sequences are always written from N terminus to C terminus. हम N से C की तरफ जाते हुए peptide chain को लिखते हैं. So for example, you are desired to synthesize serine alanine, right? This is the dipeptide or seryl alanine or allyl serine you can say. So अगर आपको alanyl serine बनाना है, so you will start with alanine first and then react with serine. So this will be N to C terminus, and if you start with serine first, then you will take serine first and then alanine, and the product will be seryl alanine. Okay. So the basic backbone of any peptide will be obviously the peptide bonds, and the correspondingly peptide pendant groups can vary depending upon the R group. Okay. So it's very important, जैसे अभी हमने bradykinin का structure लिखा, and suppose you have any biomolecule which is given to you, and suppose you want to decipher or decode its structure or its sequence, so you have to follow a certain number of the steps. Okay, it's not that you just start sequencing by using the terminal analysis which is mentioned in the syllabus. So उसके लिए कुछ steps होते हैं which one has to follow, and the first step in that process is the disulphide linkage degradation. So सबसे पहले जो भी प्रोटीन या आपके पास पॉलीपेप्टाइड है तो उसके डाइसल्फाइड बॉन्ड्स को तोड़ा जाता है. उसके बाद देखा जाता है कि उस पहली पॉलीपेप्टाइड में कितने अमीनो एसिड्स हैं और उन क्या क्या अमीनो एसिड्स हैं और उनका कितना कितना कॉन्सेंट्रेशन है. And the third step is to know that how those amino acids are arranged in a sequence. उसे हम बोलते हैं sequence analysis या terminal residue analysis. ओके अब वो रेजिड्यू एनालिसिस अगर आप एन टर्मिनल से करना शुरू करो तो उसे एन टर्मिनल रेजिड्यू एनालिसिस बोलते हैं, और अगर सी टर्मिनल से शुरू करो तो उसे सी टर्मिनल. बट बिफोर गोइंग टू द सीक्वेंसिंग स्टेप इट इज नेसेसरी to do first of all break the disulphide linkages, and the second one is to determine the composition of amino acids. Otherwise आपका sequencing का कोई मतलब नहीं बनता है, because you don't know what are the blocks and you are going to try what are in what sequence the blocks are arranged. Getting it? There is no point to it. So in the beginning also I asked you to the question that what happens to your hair when you go for the treatment processes. So actually what do? They break the disulphide linkages on your hair to the corresponding thiol linkages, and then they correspondingly फिर से दोबारा से वो treatment करके new bonds बनाते हैं. So what is rebonding actually? तो क्या वो rebonding का मतलब, rebonding नेम ही उसी से आया है कि पहले existing hairs के strands के बालों bonds को break करो, कुछ केमिकल अप्लाई करो और फिर से उस बॉन्ड को कंस्ट्रक्ट कर दो. सो जो आपके कर्ली हेयर्स होते हैं वो उस केमिकल एप्लीकेशन से स्ट्रेटन अप हो जाते हैं. सो ऑब्वियसली दैट इज नॉट अ नेचुरल प्रोसेस. यू आर प्लेइंग विद द केमिस्ट्री ऑफ ऑलरेडी existing hairs. I'm not saying that it's not safe, but yes, it should be taking into consideration the temperature. Okay, it's not that because thiol groups are the hairs are made up of proteins और वो बहुत जल्दी proteins denature हो जाते हैं, and there is a chance of burning of hair also. So coming back to the establishing the protein structure, the first step is breaking the disulphide bonds. So the first step is then the structure is to determine the protein structure is to break all the disulphide bonds and opening any disulphide linkages and separating the individual peptide chains. So individual peptide chains जब separate हो जाती हैं, suppose मान लो ये curly hair है और जब आपने disulphide bonds को तोड़ दिया तो वो straight hair हो गए. So the same thing which we are doing to the polypeptide also. अब अगर आप simply disulphide bonds को treat करके वापस से thiol linkages की तरफ ले जाओ, so I told you जब हमने reactions कर रहे थे, I told you that thiol has a capability of again oxidizing back to the disulphide. So to prevent this हम disulphide bonds को treat करते हैं परफॉर्मिक एसिड से. So advantage क्या होता है परफॉर्मिक एसिड यूज करने का, that the products are now the cystic acid residues. ये क्या बन गए आपके, ये दोनों chain A and chain B, these are the cystic acid residues, the sulfonic acid derivatives and not the thiol derivatives. Thiol derivatives इसलिए हम prefer नहीं करते because they get oxidized back to the disulphide bond. So therefore we use this and give the easily separatable cystic acid residues which can be further purified and analyzed separately. अब एक बार जब ये पेप्टाइड चेन्स अलग अलग तो हो गई पर अब हमें इन्हें प्यूरिफाई भी करना पड़ता है, सेपरेट करके वी नीड टू प्यूरिफाई देम. सो दैट प्यूरिफिकेशन इज डन ऑन द बेसिस ऑफ देयर साइज, solubility in the solvent and the charge they carry. So commonly adopted methods for purification of the chains are dialysis, ion exchange, electrophoresis and chromatography. And the next step, once they have been separated, the peptide chains are separated, purified, now the next step is to determine कि वो peptide chains किन amino acids से बनी है और उन amino acids की कितनी concentration है particular peptide chains में. So that comes to the next step which is determination of the amino acid composition. So composition निकालने का first step होता है पूरे की पूरी peptides chain को hydrolyze कर देना. So the whole peptide chain is hydrolyzed by boiling it for 24 hours in HCl. मतलब peptide chain को पूरा का पूरा chop करके corresponding amino acids में तोड़ देना. Now what you have the solution of all the amino acids which are there in the polypeptide chains. So now this solution is passed through something called as amino acid analyzer. ये है amino acid, this is how an amino acid analyzer looks like. Okay, and that the mixture of amino acids को क्या बोलते हैं जो हमारा बना था hydrolysis करके, that mixture is known as hydrolysate. So, once the mixture of amino acids has been obtained via the hydrolysis of the peptide chains, it is subjected to an analyzer which is known as amino acid analyzer, and the mixture of amino acids is known as hydrolysate. This is placed on the column of an amino acid analyzer, and once the mixture passes through this column, the various amino acids are separated depending upon their size and the charge they carry. Okay, and so the different amino acids move at different speeds because they have different, each of them has different affinity for the cation exchange column. Or the solution which emerges from this analyzer is treated with ninhydrin. So, once the solution is treated with ninhydrin, all of them will react with the ninhydrin to give you the Reumann's complex, which is the blue color complex. Now, the absorbance of each of the complex of the every amino acid will be different depending upon their structure. So, the absorption of the light is recorded and printed out as a function of the time. Okay. So, the different amino acids are separated depending upon their absorbance value because they have different structure. So, they will give a different peak in the spectra, okay, and it will be since it is a colored complex, so therefore, it can be easily observed. So, the amino acids present are then identified by comparing their retention times with the known values, यानी कि एक standard amino acid mixtures की value से उसको compare किया जाता है, and the area under each peak tells us that in how much quantity each amino acid is present relative to a standard mixture. Say for example, proline ka peak is this much and in the standard mixture it is this much. So, if it is around thrice, so you can say that proline is thrice the standard quantity of amino acids which is there in our reference standard. So, इस method से हमें पता चल जाता है क्या that how many amino acids are there and what is the concentration. Okay. So, once we have established the concentration of amino acids and the type of amino acids, we want to know that how these amino acids are arranged in the sample, and to know that we do the sequencing analysis or the terminal residue analysis, and to carry out this either it is done in the from the N terminus or either it is done from the C terminus. So, once the amino acid from the chain is cleaved, the rest of the chain is left intact. जब आपने एक amino acid को chop कर लिया, then the left of the chain is left intact and the cleaved amino acid is separated or identified and the process is repeated to the rest of the chain. So, कहने का मतलब है कि if you have an amino acid chain made, a polypeptide chain made up of X, Y, Z, and if you do the N terminal analysis, तो पहले आप एक amino acid को chop off करोगे, analyze it, then you chop off the second one, analyze it. So, thereby you can establish the sequence in which those amino acids are linked together. So, the amino acids may be cleaved either from the N terminus or C terminus. So, N terminal analysis is done via Edman degradation. Edman degradation uses a chemical phenyl isothiocyanate. So, when phenyl isothiocyanate reacts with the peptide chain in the presence of the pyridine, it forms the derivative which is known as a thiazolene derivative. Okay. So, this thiazolene derivative changes to a more stable form which is phenyl thiohydantoin derivative. So, this phenyl thiohydantoin derivative is formed for the first amino acid. That means that this chain is broken at the first peptide bond and then you get the corresponding phenyl thiohydantoin derivative. You are starting with phenyl isothiocyanate and getting the corresponding thiohydantoin derivative. So, जैसे जैसे एक आपके N terminal amino acid का hydantoin derivative बनता जाएगा, you can keep on separating it and analyzing it, still you get the whole sequence of the peptide chain. So, suppose ये आपकी peptide chain है, okay, and this is the first amino acid, that is the N terminus. You are reacting with phenyl isothiocyanate, it forms a conjugate, okay, and then it is converted into the phenyl hydantoin. Okay, this is the hydantoin of the first amino acid and the remaining chain as such. इसका आप characterization कर सकते हैं, analysis कर सकते हैं, and similarly the cycle can be repeated till you get to know the sequence of the complete chain. C terminal analysis is done by using the enzyme carboxypeptidase, where we use the enzyme to cleave the amino acid chain from the carboxylic end, that is this particular end. Okay. that is this particular end. Okay, so carboxypeptidase will keep on dissecting the peptide from the carboxylic end. So ये वाले peptide bonds break होते जाएंगे and this is where the carboxypeptidase will cleave and we will keep on getting to know the peptide sequence from the carboxylic end. इसी तरीके से दो और एंजाइम्स हैं आपकी ट्रिप्सिन और काइमोट्रिप्सिन, दे कैन ऑल्सो बी यूज टू डिसाइफर द पेप्टाइड सीक्वेंस फ्रॉम द C terminal end. So let's go for a break, and before going for the break I'll offer you a question. Suppose you have a tripeptide made up of glycine, alanine, valine, and suppose you degrade it with using the enzyme carboxypeptidase. Which is the first fragment of amino acid which you will be getting upon the degradation? The answer to this question will be given to you after the break.
Hello everyone, I am Dr. Richa Arora and welcome to S. Chand Academy. So I am going to make videos on very important topics of physical chemistry. और topics होंगे solutions, phase equilibrium, conductance and electrochemistry. इन videos में हमारा focus रहेगा basics पे और applications पे. तो ये वीडियोस चाहे आप अंडर ग्रेजुएट स्टूडेंट हो, चाहे आप पोस्ट ग्रेजुएट स्टूडेंट हो, चाहे आप इंटरव्यू की प्रिपरेशन कर रहे हो या किसी एंट्रेंस की प्रिपरेशन कर रहे हो, ये सारी वीडियोस आपके बहुत काम आएंगी. सब्सक्राइब कीजिए एस चांद अकेडमी एंड प्रेस द बेल आइकन टू गेट द नोटिफिकेशंस.
Hi. Welcome back to S. Chand Academy, and before going for the break I asked a question, and suppose that you have a tripeptide of glycine, alanine, valine and you treat it with carboxypeptidase. Which fragment of amino acid will you get the first? The answer was just discussed in the previous slide where I told you that carboxypeptidase specifically cleaves the carboxylic end of the peptide chain. So, the last amino acid in the chain was valine, right. So, the first amino acid which will be obtained will be the valine, and subsequently you can get the later on sequence. So, continuing again with the topic, we will move on to the synthesis of peptides. Okay. So, peptides are usually synthesized by a stepwise approach. Okay. Suppose we want to synthesize a peptide chain, say AB. Okay. अब उसके लिए हमें दो अमीनो एसिड्स की ज़रूरत है A and B, right? Suppose you start with a mixture of A and B, so what are the possibilities? आपने A से स्टार्ट किया, आपने B से स्टार्ट किया. अब दोनों के पास ही N टर्मिनस है, दोनों के पास ही C टर्मिनस है, that is both of them have free amino and the free carboxylic group. So when you try to make a peptide out of the two, what are the possibilities? You can get AA, right, you can get AA, you can also get BB, and you can also get AB, and you can also get BA. Lot of difference. What we wanted is just AB, but the possibilities are much more. You can get a mixture also. So to avoid the formation of a mixture, we have two methods and specific chemical reagents which can precisely give us the desired result. Okay. So the two methods are based upon the solution phase chemistry and the solid phase chemistry. The solution phase method involves adding reagents to solutions of growing peptide chains and purifying the products as needed. So this is obviously carried out in the solution phase. जबकि जो solid phase method है, it involves adding reagents to growing peptide chains bonded to a solid polymeric particle. So इस केस में एक solid polymer यूज़ किया जाता है जिस पे आप amino acids को बॉन्ड करते जाएंगे. सो कहने का मतलब ये है कि हम एक तरफ से तो हम रिस्ट्रिक्ट कर लेते हैं. सपोज ए बी चाहिए तो पहले ए को पकड़ लिया एक साइड से और उस पर और अमीनो एसिड्स ऐड करते गए. और अगर आप सॉलिड फेस कर रहे हैं तो पहले हम बी को होल्ड कर लेते हैं. यूजुअली कन्वेंशन ये है कि अगर सॉलिड फेस कर रहे हैं तो पहले सी टर्मिनल वाला अमीनो एसिड हम सॉलिड सपोर्ट से टाई अप कर देते हैं एंड देन वी कीप ऑन एडिंग द रिमेनिंग अमीनो एसिड्स. So the solution phase methods work from left to right, as I just said, that is works from N to C. Okay. So the first step is protection of N terminus. देखो अगर आप N को protect नहीं करोगे तो there are very much chances कि A जो है वो अपने ही कार्बोक्सिलिक ग्रुप के साथ कंबाइन कर जाए, उसी का NH2 उसी के कार्बोक्सिलिक ग्रुप के साथ. So to prevent that, the first step is protection of N terminus, and the protection of N terminus is done by using benzyl chloroformate as the reagent, and benzyl chloroformate is treated with an amino acid. The product is corresponding benzyl oxycarbonyl of that corresponding amino acid. So once this is now N protected, so ये आपका हो गया N this is your N protected amino acid. ये आपका N protected amino acid हो गया. Then N protected amino acid is to be reacted with second amino acid. सेकेंड अमीनो एसिड को रिएक्ट करने से पहले हमें इसके कार्बोक्सिलिक ग्रुप को एक्टिवेट करना पड़ता है. एक्टिवेट करना पड़ता है ताकि जो आपका सेकेंड अमीनो एसिड आ रहा है वो ही कहीं अपने ही कार्बोक्सिलिक ग्रुप के साथ रिएक्ट ना कर जाए, उसी का सेल्फ कंडेंसेशन ना हो जाए. सो वी नीड टू एक्टिवेट आवर अमीनो एसिड सो दैट the incoming amino acid reacts with this only. So for activating we use various reagent, and one of such reagent is ethyl chloroformate. So the corresponding product is anhydride of the carbonic acid, and once it has been activated, okay, so this is your N protected C activated amino acid. ये आपका N protected and C activated amino acid है. So one is our N protected C activated amino acid. This can now be reacted with the second amino acid to give the corresponding N protected dipeptide. Okay. Suppose you want this reaction till dipeptide only, तो आपका काम क्या रह जाता है, सिर्फ इस N protecting group को remove करना, and this N protecting group can be easily removed by treating it with the corresponding reducing agent such as hydrogen and palladium to give the corresponding dipeptide. So, okay, so you can get the product till the desired stage. We wanted it till this stage only, alanine valley, right? The N protection can also be done by using tertiary butyloxy carbonyl group, which is also known as the T-Bog group. Then the second method of synthesizing the peptides I told you is the solid phase method, also known as the Merrifield Solid Phase peptide synthesis, and this is a solid phase synthesis which is done in the opposite direction. जैसा कि मैंने आपको बताया, that is it is done from C terminus to N terminus, and in this the C terminus वाला amino acid is bound to a solid support. तो इसमें होता क्या है, जो C terminus वाला amino acid है हम उसको एक solid support से tie up कर देते हैं, and that solid support is a polymer made up of styrene. Okay, so this is basically a polystyrene derivative, and it is made by using styrene and parachloromethyl styrene. Okay, so once this resin is obtained, we want to now attach the C terminus of the desired sequence to this resin, और वो अटैच करने के लिए भी हमें पहले उस अमीनो एसिड का नाइट्रोजन यानी कि अमीनो ग्रुप प्रोटेक्ट करना पड़ता है. सो फर्स्ट यू प्रोटेक्ट दैट अमीनो एसिड एंड देन यू टाई अप टू दैट रेजिन. सो दिस इज योर N प्रोटेक्टेड एंड C बॉन्डेड अमीनो एसिड. ये आपका लास्ट अमीनो एसिड है चेन का. ओके, देन here the protecting group used is Bock group, tertiary butyloxy carbonyl group, which is introduced by using di-tertiary butyl dicarbonate, right? So this is your Bock amino acid. So once this has been synthesized, it is reacted with the resin, and एक बार ये resin से attach हो गया, फिर हम Bock group को remove करते हैं using trifluoroacetic acid. So what do you get? You get the free amino acid which is now bonded to the styrene. Okay, अब आप क्या करेंगे, then you want to couple the first amino acid. अगर डाइपेप्टाइड बना रहे हो, एक अमीनो एसिड आपका रेजिन पे अटैच हो गया, अब आप दूसरा यानी कि चेन में फर्स्ट वाला जो अमीनो एसिड है उसको अटैच करोगे. सो दैट इज डन बाय यूजिंग द कपलिंग रिएजेंट विच इज DCC. सो सॉलिड फेज मेथड में जो कपलिंग रिएजेंट या आप कहो कि C एक्टिवेटिंग रिएजेंट जो है वो होता है आपका DCC, this which is dicyclohexyl carbodiamide. Okay, so when DCC reacts with the carboxylic end of the second amino acid, it activates the carboxylic group, thereby it can react with the amino group of the amino acid which was attached to the polystyrene group, and the product of this reaction is the corresponding amide जो हमारा dipeptide होगा, and the byproduct will be DCU यानी कि dicyclohexyl urea. So overall process अगर हम देखें तो it will be, आपने start किया N protected amino acid, suppose you wanted a dipeptide say AB, so पहले आपने B को resin से attach किया, you started with the resin, you reacted it with ZB, ये आपका N protected second amino acid है, so this will be your ZB resin, and then what you did, फिर आपने Z को remove किया using trifluoroacetic acid, so this is B resin, and then in the presence of DCC you coupled it with A, so this is A B resin plus DCU, right, and then A is obviously आपका N protected होगा, right, A is also N protected, so this is ZA, so इसमें से भी आप जो है Z को remove करोगे, resin को remove करोगे, so you will get the corresponding dipeptide AB. So basically ये जो sequence है, it has worked from B to A, right? तो अगर आपको change चाहिए say A, B, C, so you will first start with C, then you will add A, and then you will add B, and then you will add A, C to N terminus. So this is how the solid phase peptide synthesis works, and the resin is removed by washing it with HF. Okay. Now the last part of the topic is protein structure. So protein structure basically they can exist in three forms: primary, secondary and tertiary. Primary structure means the amino acids are just aligned linearly, right? So they form the basic components of various polypeptide chains. So जहाँ पे भी amino acids linear fashion में present होंगे उन्हें हम बोलेंगे primary structure. Secondary structure means that which are formed within a polypeptide due to the reaction between the atoms. So there are two forms in which secondary structure of proteins may exist: alpha helix and beta pleated, and both of them are held together by hydrogen bonds. जब हमने peptides शुरू किया था, I told you that they are linked together by covalent bonds, that is peptide bonds or disulphide linkages. But अगर दो peptide chains आपस में linked होंगी, so they will be linked together by electrostatic interactions called as hydrogen bonds. So alpha helix will be helical in shape like this, and beta pleated में sheets होती हैं. These are the sheets which are linked together in the linear fashion, which are bonded together by electrostatic interactions by the CO-NH bond. देखो तो CO एक peptide chain का दूसरे peptide chain के NH से electrostatically interact कर रहा है. इन दोनों के बीच में एक hydrogen bonding है, not a covalent bonding. Tertiary structure is a three-dimensional structure where there are two sorts of interaction. One is due to the interactions between the R groups, ये hydrophobic interactions होती हैं. दूसरी होती हैं आपकी ionic interactions which is between the various peptide bonds. Okay. So whenever there is, these are the ionic interactions which are the peptide bonds which of one linking with the peptide bond of other, that is the hydrogen bonds. The other ones are hydrophobic interactions. So hydrophobic interactions interior में होती हैं और hydrophilic interactions they form the backbone, they are on the outer side, okay, which makes the peptide soluble in the solution. The last type of the structure in which the proteins may exist are the quaternary structure. They are quite tense structure, and the most common example is hemoglobin. So when multiple peptide chain subunits come together, they form the quaternary structure, and usually these type of the structures form the functional biomolecules like hemoglobin. So जैसे कि hemoglobin में it is linked to the imidazole ring, is linked to the nitrogen atom, and then that contributes to the transport of oxygen throughout the body. So usually the quaternary structural proteins are functional molecules. With this we end our topic on the proteins and amino acids. So that's all about this video. I hope you like the lecture, and for more information regarding our lecture you can refer to our book by S. Chand Publishing, the link to which is given in the description box below. Don't forget to like, share and subscribe to our channel, and stay tuned for more videos, and keep watching. Thank you.
All rights reserved. This video has been prepared for educational purposes only. No part of it may be reproduced or copied without the permission of the copyright holder.